Welcome everybody to AGS, the Arthropod Genomics Symposium X, session number 2, Insect Genomics, Biology and Evolution, presented by or led by Lindsay Perkin. So this is a joint effort between the USDA, ARS, National Institute of Health, NIH, as well as the I5K, the International Sequencing Consortium, aimed at sequencing 5,000 arthropod genomes. We've already conducted session 1, and again, we're approaching now, and happening now is our session 2. And take note, please, of the upcoming session 3 and 4. And the QR code can be scanned right off the screen. And I'll drop that into the chat later on so you can check out the information on our upcoming sessions as well. So this is all made possible, just not by those sitting in front of you today, but a lot of people working in the background made this possible. We had our administrative support by our national program leader, Kevin Hackett, myself as lead organizer, Glenn Hayes working in the background, getting all the technical aspects for the Zoom and the registration forms, Anna Childers doing the website and Twitter, Pia Olsen, does a great job of keeping all the videos in line and playing nice and smoothly, Rob Waterhouse, who's worked to update and keep the Slack channel up to date with all the pertinent information. So thank you to everybody. So, and again, we have the additional resources mentioned previously. The website is present there with the link. And again, all this information will be dropped into the chat for everybody to click on. The registration form for the additional sessions, if you have not done so already. And all these are being recorded, and these sessions will be posted on the YouTube channel with the link there. And again, will be posted later. And please go ahead and check them out on the different playlists. And that Slack channel is present right there. And that's the link for joining if you have not already to require a registration and an approval. So that'll happen rather quickly. So at this point in time, I'll turn it over to our moderator, Lindsay Perkin.
Hi, thanks, Brad. Like Brad said, I'm Lindsay Perkin. I'm with ARS in College Station. And I'm really pleased to show the lineup of speakers we have today. Every year I do this, and I also organize a lot for plant animal genomes. I always worry what kind of speakers we'll end up with, and I always surprise myself every year in finding fantastic speakers. So thank you, speakers, for agreeing to be part of this symposium. It really what makes this a successful virtual meeting. All right, so one last thing I need to mention is that when you, as the audience, want to ask a question, don't hold back and just put those questions into the Q&A. That's preferred over the chat to make sure that I see it and can read it for our speakers later. And in this webinar, the talks are actually recorded, but then we'll open up the floor to live questions with our speakers after. All right, so without further ado, I want to introduce our first speaker of the day, Dr. Kirsten Verster. Her interests are in how functional genes evolve in unique ecological contexts. She got her PhD at the University of California, Berkeley, and she's currently a postdoc at Stanford University, doing some really cool horizontal gene transfer work. So Pia, if you'll cue up the first talk, please.
Hello, my name is Dr. Kirsten Isabel Verster, and I'm so excited to talk to you guys today about my PhD dissertation research, which I titled A Toxic Triangle, or How Horizontal Transfer of Toxin Genes Protects Insects Against Parasitoid Wasps. Talk is pre-recorded, so I don't necessarily know what else we've spoken about today, but my guess is that a lot of the different talks deal with more traditional type of inheritance known as vertical inheritance. A lot of the theories of evolution, the way that things have been considered for the vast majority of time we thought about genetics has been dealing with parent to offspring. So the example I'm going to show is that of my mom, who's very beautiful, and my dad, who's very charismatic. And they combine their DNA and pass it down vertically to me, who is a perfect combination of both of those traits. This is known as vertical inheritance, or the passing of genes down from parent to offspring. What I'm going to be talking about today is horizontal inheritance. And the example I'm going to use is that of an alligator, because I'm from Florida and we need to call it out. So what would happen if an alligator were to exchange its DNA with me? You might observe that I acquire some different traits. For example, maybe I develop really scaly skin or sharp teeth. This would be known as horizontal inheritance, or the passing of genes between different species. This has been a relatively well-known phenomenon in bacteria for a long time, but more recently are we realizing that it is also abundant in other aspects of the animal kingdom as well. So the first major question I'm going to tackle is, do Drosophila have horizontal gene transfer? There is a compelling reason that I believe this. The past 10 years have seen a huge surge of studies in insects that have enabled us to realize that a great deal of adaptations have been facilitated by horizontal gene transfer, usually from bacteria or from viruses. My personal favorite examples are those of ticks, for example, which acquired anticoagulatory genes from bacteria, which means they're able to keep sucking blood without the blood coagulating and hardening, which means they can't drink it anymore. Additionally, there are stick insects, which acquired enzymes from bacteria that help them digest plant cell walls. And the next one is going to be Drosophila. In this case, one specific type of fly called Scaptomyza flava, which is in the Drosophila group. So this fly is pretty interesting because unlike most Drosophilads, which feed on dying, decaying material, Scaptomyza flava feeds on fresh mustard plants. You can see it sucking up plant juices right there. And the reason that we decided to interrogate their genome to see whether or not maybe there was horizontal gene transfer in this group is because we were thinking maybe there was something similar to the stick insect cellulase example. What if horizontal transfer from a bacteria enables some sort of adaptation that helps Scaptomyza flava feed on fresh material where most Drosophila can't? Something that could have spurred a very rapid adaptation to this new unexploited niche when it comes to Drosophila. So that was the motivation for us searching for the genome. But how do we look for horizontal gene transfer in the genome, you ask? Well, that's what the next slide is about. The way that we detect horizontal gene transfer is through conflict, or as I like to say, drama. Specifically, the type of conflict we're looking for is phylogenetic conflict. Phylogenetic conflict is a discrepancy between the species tree and a gene tree. The tree that we see on the left is a species tree, which recapitulates evolutionary history of the species. So when we look at this phylogeny, we know that humans and chimpanzees are more closely related to each other than they are, for example, to a mouse or to an iguana. Gene trees, on the other hand, don't necessarily reflect the species histories. So one example that I think is super interesting is that of prestin, which is a gene that plays an important role in echolocation. If you look at this prestin phylogeny, you might think that bats and dolphins are more closely related to each other than they are to other bats, for example, or whales, when we know inherently this is not the case. This is one example of phylogenetic conflict, where the gene tree does not look necessarily like the species tree. But because of similarities in the function and structure of the gene, they do seem to be placing the actual species that hold the gene more closely related to each other. So if I were to change the species tree on the left, for example, I might instead see that there are two tips in unexpected places. In this case, I might see a human tip located next to an iguana tip. This right here is an example of conflict. We don't expect humans to be more closely related to each other than they are to a capuchin, for example. If I were to think of this from a horizontal gene transfer approach, my thought might be at some point, maybe iguana DNA got passed down to humans or vice versa. Of course, there's a lot of things to consider before you instantly assume either A, that there is a similar structure or function of these genes, or B, horizontal gene transfer. One of the major things that plague studies of horizontal gene transfer is the threat of contamination. DNA is small, DNA gets in everywhere. So it makes sense that you need to be very certain about the provenance of a gene before you automatically assume that it comes from somewhere different. Something very common in studies of horizontal gene transfer is that maybe you think it's a bacterial transfer, but it could just be bacterial contamination. So when I was looking for horizontal gene transfer of the gene I'm going to talk to you about, there was a lot of things that I did to make sure that it was real and that it was not a contaminant, because I didn't want to spend 2 years of my PhD studying all this only to realize it was something floating in the air. Some of the things that I did were I looked at coverage depth analysis to make sure that the reads were distributed equally, or what you would expect from other eukaryotic genes on a scaffold. So, for example, if it were bacteria, in any given cell, you would have, for any given period, like amount of space, you would have a lot more bacteria that could fit into that space, and there's a lot more bacterial DNA than you could eukaryotic DNA. So you would expect some unevenness in the read distribution. But we did this analysis and it was not the case. Additionally, you look for synteny of the group. For example, if it's located on the same scaffold with two neighboring genes across a variety of different species, it's more likely to be real because you don't expect that pattern to happen by accident. You would only expect it to happen by some initial horizontal gene transfer event, then proceeded by vertical inheritance. Additionally, there were other things that we did, such as making sure that the scaffolds were long and they encode other eukaryotic genes. So I searched to scaptimize the flava genome, expecting there to be transfer of some genes responsible for plant digestion, but that theory turned out to be entirely wrong. However, I did find transfer as something else that was very interesting and led me on a totally unexpected path. The genes that I found were transferred were cytolethal descending toxin B, which is a DNase, hence why there's a picture of a scissor chopping DNA. CDTB is traditionally known to be found in pathogenic bacteria, where it can cause cellular dissension and apoptosis, leading to cell death. CDTB is one of the major toxins implicated in irritable bowel syndrome because it destroys the cells lining the gut. The other gene that we found transferred was apoptosis-inducing protein of 56 kilodaltons, or AIP56. This is a peptidase, and it's largely traditionally known when secreted by fish pathogens, and like CDTB, also causes apoptosis, albeit of fish host blood cells. So I'm going to tell you first a little bit more about CDTB and the approach that we use for CDTB and how we discover a transfer. Like I mentioned, it does cause cellular dissension and apoptosis. So you can see in this image, this is images of a healthy cell, but when intoxicated with CDTB, what happens is the cell will start to try to mitose, starts descending, but then it is not able to properly divide, which then leads to cell death. When we constructed a phylogenetic tree of CDTB, we expect in cases of horizontal gene transfer, we saw that there were a lot of cases of animals or organisms that were very distinct from each other being placed together on the phylogeny. So we found CDTB encoded in the genomes of a variety of different insect groups, including aphids, Scaptomesa flava, which I mentioned earlier, another group of flies called the Ananaceae subgroup flies, which are also Drosophila species, and some species of Lepidoptera, which includes moths and butterflies. And these insect groups were located next to CDTB sequences of bacteriophage and also bacteria. But the thing that were interesting about these species is that, for the most part, they were known endosymbionts, or organisms that live within the body or cells of other organisms, usually in some sort of mutualistic sense. So the reason that's pretty interesting is that it suggests that there was horizontal gene transfer between organisms living next to each other. And one of the major theories I've seen put out about how HGT operates is that it is facilitated by closeness. So you have more opportunities for ruptured genetic material to access the germline if it happens to be present very frequently, which you would expect in an endosymbiotic context. When I look at the different times the species diverged and when there were species that don't have it versus those that do, this puts the first estimate of CDTB transfer as about 40 million years ago, which is quite a long time ago. It's very cool. Because bacteria that secrete CDTB have important medical implications, we have a pretty good understanding of the different amino acids and how they function in this capacity. Thus, for CDTB, there has been a lot of site-directed immunogenesis done on specific amino acids, so we have a good sense of how changing these affects the function and the toxicity of the protein. Even though insects and bacteria are very different, we observe that a lot of the amino acids that are important in these enzymatic activities or these DNase activities are almost 100% conserved even between these very phylogenetically distinct groups. We see that this conserved structure also has manifestations in conserved function. So we actually purified insect CDTB, in this case specifically CDTB from Drosophila ananaceae, and we incubated it with supercoiled plasmid, which is plasmid that's just very, very tightly coiled, and as a consequence, it will go lower down in a gel than you would expect a normal plasmid to go. And what will happen is that when incubated with DNases such as CDTB, that the plasmid is going to get nicked, which is going to make it bigger, which means that it'll float up higher on a gel. So as you can see in this image over here, when we add increasing quantities of CDTB protein, we see more of what are called the relaxed and linear species of the plasmid, suggesting DNase activity. So this was a sort of experimental, like artificial situation where we actually do see this DNase activity at work, which we found very exciting. In order to understand a little bit more about under what context CDTB is important, we checked expression of CDTB across different life stages in two species, in Drosophila ananaceae and Scaptomyza flava, the leaf-eating fly. And in both species, we found that it was most highly expressed in the larval stage. This suggests that the gene is more important during this life stage. Now I want to tell you a little bit more about AIP56, one of the other toxin genes that we found in these Drosophila species. Like I mentioned, it also induces apoptosis, hence its name, apoptosis-inducing protein. And like I mentioned, it is found in fish pathogens and it causes apoptosis of their blood cells. So there's a kind of gruesome image over here showing that it does cause massive die-offs in aquaculture environments because it's secreted by this bacteria called Photobacterium Dancellus subspecies piscicida, and it leads to destruction of blood cells, and you might see that they have like enlarged spleens and other structures in fish. As with CDTB, we also saw a variety of different insect lineages that encoded it, including a lot of different Lepidoptera species, again, some wasps, and Drosophila, specifically the Ananaceae subgroup that previously had CDTB. As with CDTB, they are also located next to endosymbiotic phage in this case. And so basically what we're seeing is that both of these different groups of toxins seem to possibly have been acquired by endosymbiotic phage or bacterial donors, but in particular, the donor ancestor that may have donated these genes is pretty unusual for the reason I'll tell you right now. And while it's interesting because CDTB and AIP56 are generally known to be secreted by pathogenic bacteria, and they kind of exist in this bad, very negative space for eukaryotes. But there is one really interesting example where they actually have proven to be beneficial to eukaryotes. That is in the pea aphid, Asterothosiphon pisum. A pisum has beneficial bacterial endosymbionts known as Hamiltonella defensa. And Hamiltonella defensa has encoded in its genome a bacteriophage known as APSE, or apism secondary endosymbiont. How does this relate to CGTB and AIP56? Well, interestingly, encoded in AIP and APSE are these two toxin genes that I mentioned earlier, immediately next to each other. And these specifically, this is one strain of APSE that has them next to each other called APES2. And the reason that APES2 has been shown to be beneficial to endosymbionts is in this one context when it comes to parasitoid wasps. So parasitoid wasps, you can see that over here they're attacking an aphid, and what happens during that attack is it'll oviposit an egg into the aphid, and then what will happen is that this egg will hatch into a little baby wasp, and then that baby wasp eats the aphid body alive until at the very end a wasp will come out, leaving the mummified body of the aphid behind. However, what happens in the presence of this apes encoding these toxins? Instead, we see that a wasp will lay an egg, but for the most part, the wasps do not survive. So in this one interesting, unusual context, it seems as though these toxin genes are actually protecting the P aphid, even though the mechanism is very unclear. Wasps are abundant in the animal kingdom, and they attack a wide variety of insects. This is a cool image I just wanted to share of a parasitoid wasp attacking a Drosophila larvae. And they do, in fact, attack Drosophila. You can see here in this image that a Leptopolina wasp is palpating the ground and it's ovipositing into the larva that it finds inside the media. So with that, the answer to the first major question, do Drosophila have horizontal gene transfer? Yes. I didn't search all Drosophila for all gene transfers, but I did identify two toxin genes. It's called CGTB and AIP56, which may have been inherited by endosymbiotic organisms. And it seems as though it's highly expressed in larvae, has conserved DNA structure and function, which leads us to the next question. Well, what does it do? Do these toxin genes protect Drosophila, like aphids, from parasitoid wasps? That's what I decided to look into. And what I decided to do was look at one specific type of Drosophila species. Like I mentioned, these two genes protect the aphids from parasitoid wasps, albeit in a kind of convoluted or kind of complicated way, where they protect them because they're embedded in a secondary endosymbiotic genome, which is part of the endosymbiont, which is existing inside the cells of the P aphid. It's a little bit complicated, but these genes were transferred to another organism, specifically Drosophila ananaceae, both of them. And this was great because, according to Dr. Singh, Drosophila ananaceae is a good model species for genetical, behavioral, and evolutionary studies, which is perfect. This person is obsessed with ananaceae and has a new study on them every year. It's very cool. But they're not only cool for these reasons, but also because of how these genes were inherited. So CGTB is located immediately upstream of a fused version of CGTP with these AIP56 genes, specifically what's called the B component of the AIP56 genes. And then that was duplicated. So not only was there this horizontal gene transfer, there's also this duplication. And the thing that's interesting about Drosophila ananaceae is that they're actually really, really good against parasitoid wasp attack. In a lot of different studies, when you can have a whole swath of wasps attacking a whole swath of ananaceae or a whole swath of Drosophila, ananaceae tends to have the highest survival rates, which is really interesting. Drosophila ananaceae, additionally, also has a very cool type of cell called multinucleated giant hemocytes. These giant hemocytes are very adept at attacking or crawling toward parasitoid wasp eggs and attacking them via unknown mechanisms. So the hypothesis I was asking myself is whether or not these two genes might possibly be implicated in these multinucleated giant hemocytes, or if they play a role in some capacity in the amazing parasitoid wasp defense that NNSA has. In order to address these questions, we had a collaboration with an amazing group of scientists at the Biological Research Center in Hungary, two major players being Dr. Gyöngy Szanegi and Dr. István Andó. They're very adept at coming up with these different, at creating antibodies that enable us to visualize where proteins are located in the tissue and the organs of Drosophila ananaceae. And so what we did is we identified, we created a whole swath of, created a whole swath of immunohistochemistry experiments that helped us identify where these proteins are actually located across the developmental stage, across different cells or tissue types, and after wasp attack. This was a much more comprehensive assay than what I did previously when I was just looking at qPCR of these different life stages of the flies. And so we found that CDTB was very highly expressed in embryos, which is kind of surprising. This was something I hadn't tested before at all, suggesting it plays a really important role in embryonic development. That additionally, it was most highly expressed in the macrophages and fat body, which is an organism that is important in metabolism and also the immune system. And we saw that it was slightly induced following parasitoid wasp attack. The two fusion proteins had different patterns of expression. So we saw that they were both most highly expressed in larvae. They were very highly expressed in the fat body, and they were also very highly induced following wasp attack, which was it suggested that these play a very important role in the wasp defense. I want to show this really beautiful image that they generated of parasitoid wasp larvae that were laying an egg, that laid an egg and hatched inside Drosophila ananaceae. The stuff that is green in this image is actually the CDTB AIP56 fusion protein. And these wasp larvae are dead. And it seems as though they're kind of enveloping that the protein is enveloping them in a kind of toxic hug that may have contributed to their death. So I thought this was really interesting, and we had these really great immunohistochemistry images showing where the proteins are actually being expressed on this very fine scale level, which led me to the next question. I wanted to actually see what happens if I remove the toxin. And so what I did is I used CRISPR-Cas9. We got Drosophila ananaceae to lay a whole bunch of eggs, and then we injected Cas9 and guide RNAs targeting all the different genes. Like I mentioned, there were 3 different genes, and it took forever because Drosophila ananaceae is not a model system, so we didn't have a lot of the tools you might use to do this very quickly in Drosophila. So I went for a very simple approach. I just wanted to create a knockout in each gene in some kind of modular fashion. And this involves having them lay eggs, and then once they hatched and they were adult, I would very carefully cut a leg, and then I would quickly do DNA extraction from it and then PCR, and then submit it to be sequenced, and I would look at the sequence, and then I would try to get multiple flies that had the same genotype to mate until eventually I would have an entirely homozygous line of these flies, which eventually I did, which I was thrilled about, and I ended up with 6 different lines of the variety of iterations of the mutations, where CDTB was there, or it was present, absent, and I was very pleased with this. Afterward, what we did is that we had, again, these Leptopolina wasps, parasitized Drosophila larvae, the mutant flies in addition to the wild-type flies. And what we found was very interesting. First of all, we did take both sides of the coin. So when you're doing this kind of study, you can consider how the flies are surviving, but something else that tells that's a different side is that sometimes you need to look at the wasps as well. Because if you have all the flies survive and no wasp, it tells you one thing, but what if none of the flies survive and all the wasps emerge? Or what if none of the flies survive but none of the wasps emerge? This tells you really how successful each side of the parasitoid host system is doing. So we had them attacked by a few different parasitoid wasps, which employ different strategies for their parasitoid attack. Leptopelina heterotoma, which employs a more like free-floating, himulent, like more active sort of defense. We saw that the wild-type fly had the most flies emerging, and we didn't really see any difference between the wasps that emerge from the different lines. So you can see here, this over here is like the wild-type fly, and this at the end is the triple knockout, which had both all CDTB and the fusion proteins removed. And even though this is not significant, we see that there's does seem to be more wasp emergence from this final group. Leptopelina victoriae, none of the flies survived, which was pretty interesting. But we did see that in the wild type, very few wasps emerged compared to the triple knockout, where we did see a lot of the wasps manage to escape or to grow to development in the triple knockout flies. And in Leptopelina boulardi, we saw that yes, there were more wild type flies that seemed to survive. But as you can see over here in this image, the reason that I had this little cross sign is because apparently, according to the Ando lab, they had never seen these wasp species emerging from this fly before. So I was very pleased that this triple knockout managed to lead to this like unusual survival of the wasp. You might be very discerning and thinking to yourself, well, maybe these flies just aren't any good. Maybe the triple knockout has reduced their fitness overall, and what we're seeing isn't actually anything about the wasps themselves, but just because they're not doing well. But when we conduct this exact same experiment with the different mutants, and we remove the larvae and we treat them and we kind of poke them a little bit, but we don't like induce their immune system with the wasps, we do see that there's no statistical significance between different lines, suggesting that the response we see is specific to their defense against parasitoid wasps. So the answer to the second major question, do these genes protect Drosophila like aphids from wasps? And the answer does seem to be yes. Based on our CRISPR experiments of loss of function, flies without these genes seem to fare more poorly against parasitoid wasps than do the wild-type flies. So in maybe a year, I hope that you have a few takeaways from this presentation, which are that these two toxin encoding genes called CDTB and AIP56 were horizontally transferred into a variety of different insect lineages most likely from endosymbiotic bacteria. And they seem to work in concert to protect flies from parasitoid wasps, which is pretty interesting because it's sort of flipping the script on how these genes worked. They originally, or in a traditional sense, are known to be bad for eukaryotic cells, but here we're seeing that they're actually being deployed to protect those same cells they evolved to destroy. With that, I would like to thank my collaborators, particularly everyone at the Biological Research Center, without whom it would have been very difficult to accomplish my PhD, so I'm very grateful to them. Additionally, of course, my PI, Dr. Noah Whiteman, and just countless others who made this project turn into something very random, just looking for gene transfer, thinking we're going to find plant cell wall digesting genes, into something totally different that delves into questions of immunity and protection and help coming from unexpected places, really. In particular, I would like to shout out Rebecca Tarnapol, who inherited the project after I left, who's been taking the system in entirely new, amazing directions. So she managed to actually do a gain of function mutation in Drosophila melanogaster. So she put CDTB into melanogaster, and melanogaster acquired the ability to get better defense against wasps, which is something we were talking about years ago that never seemed possible, but she's managed to make it come true. So with that, thank you so much for your attention. I look forward to hearing your questions. My apologies for any erraticisms that may have happened during the recording. I'm not quite used to this format, but I'm thrilled to be here, and I'm excited to hear from you later. Bye.
Thank you, Kirsten. People are being shy currently, but I have a couple questions for you, and I also got a few via text that I'll read for you to share with the group as well. So my first question, and I may have missed this, but your collaborators in Hungary, when they looked at genes post-infection with wasps, were they doing qPCR as well, or did they maybe do like transcriptomics to get like a...
Yeah, no, so they did do like those results were qPCR. We do also have like some transcriptome data, basically there's a few data sets all over the place. So there does exist some transcriptomic data for like these different life stages, but of course we're interested in specific tissues. So this little table that I showed was a summary of both qPCR data and also protein expression data. So there's lots of tables and lots of graphs I could have showed, and so I was just trying to simplify it, being like, yes, highly expressed in the larvae, yes, highly expressed in these specific tissues. But the transcriptomic data that does exist from other sources, from other people looking into other aspects of the Drosophila development, does corroborate this as well.
A related question. So presumably there are Drosophila species that also can protect themselves against wasps that don't have these horizontally transferred genes.
Absolutely.
So do when you compare their response and the response to these flies, see if I can ask this question without being really confusing here. Does Ananassi also express all those immune genes, like fly immune genes, or because they have these horizontally transferred genes, is that dampered compared to?
Yes, that's a great question. And the thing is that Drosophila ananassae, so there's like a well-known robust immune response in Drosophila melanogaster called like I want to say like lamellocytes. They have like a specific cell differentiation, like a differentiated cell which has like a known mechanism of action by which it attacks parasitoid wasp. But Ananassae does not have that. So my understanding is that there are some commonalities, but even within the Drosophila group, there are certain cell lineages that only exists in specific species. And Ananassae subgroup does not have lamellocytes, which is like a pretty well-known mechanism by which a lot of Drosophila attack cells or attack wasps.
Right.
So there's a lot of difference. There's a lot of difference, and like of course, you know, I'm sure it's not entirely. It's not like this one specific horizontally transferred gene isn't the only piece of the puzzle as to how immunity works. I know it's like it's kind of like a moving target. There's a lot of complex elements that go into it, but the point is that I think it's one of the pieces. I think it's an important piece, but it's very interesting. And just like doing this project, realizing how much diversity there is in immune strategies.
That is really cool.
People in a group.
Yeah, Ryan, go ahead and you can ask your question.
Hey, Kirsten, really cool. I was just wondering about Ananassae in general. They just kind of like a weird fly, because don't they also have a bunch of like Wolbachia incorporated into their genome? And I was wondering if there's something about them in particular, or like transposable element activity maybe is increased or something in that species.
Yeah, no, that's a great point. Yeah, no, so Ananassae, when you look up like Ananassae horizontal gene transfer, one of the first thing that comes up is going to be that like there's a whole Wolbachia genome that's been integrated into its genome. To the best of my knowledge, this specific toxin gene doesn't necessarily seem related to Wolbachia. It's not encoded on that in that area. But yeah, it's possible that Ananassae seems to be that the genome seems to be very permissive of foreign elements. But it's just interesting. Like I mentioned, this person, Dr. Singh, he has like a new study on Ananassae every single year, and he's always in his papers. The introduction is always like, this species is amazing. But I found the species a little bit annoying to deal with because their generation time is a lot longer than Melanogaster. So I had to plan my experiments on like the month scale.
Yeah, very cool. That's interesting that some species are more willing to accept foreign DNA than others. It's a whole
Yeah.
nother question that would be cool to look at. We did get one question in the chat. David Heckel asked, what is it about these toxins that make them specific to kill wasps and not like attacking cells in the host fly?
So that's a great question. That's something that the future PhD that the person who inherited the project is now working on. So interestingly, it turns out that the fusion genes, basically, if you express the fusion AIP56 genes constitutively, it does cause the insects, or like just like Melanogaster will not do well overall. But when they're expressed specifically in immune cells, they actually are going to be able to attack and like have better parasitoid resistance. But I think it's a great question that like the actual mechanisms we don't really know. There does seem to be something specific that is getting triggered when the immune system is triggered, specifically when parasitoid attack is being triggered. But it is the kind of thing where we're like, oh, this thing is a DNA, so targets eukaryotes. You would think that part of the major barrier to is getting integrated into the genome is that we need to make sure it doesn't kill the organism that's accepting it. So I think it's some sort of like taxa-specific specificity that's only getting triggered in like this specific immune context.
Cool. All right, I have one last question that was texted to me by a colleague. All right, this is a methodology question. So phylogenetic discordance was used for HGT ID, and this is also sometimes how you infer selection. So is there a particular way you curated against rapid evolution of endogenous genes versus horizontally transferred genes?
Well, in this like rapid evolution of endogenous genes, so this gene is not like my apologies if I'm not understanding the question correctly, but this gene is not like that. I think that like rapid evolution of endogenous genes, you might see that more in a gene that's present in various different members of the group. But this is only found in this one specific subgroup. And when you phylogenetically place it, it's sister to a bunch of bacterial taxa. So my impression is that just by virtue of the fact that it's like in this like large bacterial clade, I don't necessarily think that applies. And my apologies if I didn't understand that correctly.
There's no text back, so I think you answered.
Okay.
Okay, so in case you're too shy to write, or you're trying to get the courage to write a question, even though we're done with Kirsten's talk, it's not too late. We can always get back to stuff at the very end as well. But I think in interest of time, we'll go ahead and move on. So thank you, Kirsten, for a really
Thank you.
fantastic. Yeah. Okay, our next speaker today is Dr. Ryan Bracewell. His main interests are about how do new species form and understanding underlying genetic and genomic changes that arise to the evolution of reproductive isolation. He did his PhD at the University of Montana, did a postdoc at the University of California, Berkeley, and did a short stint with the USDA Forest Service as an entomologist, and he's now an assistant professor at Indiana University in Bloomington.
Hello, everybody. My name's Ryan Bracewell, and I'm an assistant professor at Indiana University. And today I'm going to tell you about using genomics to explore the sex chromosomes of beetles. So as many people here probably know, chromosome number varies pretty wildly in insects. So in some groups like flies, there are just a few chromosomes. But in other groups, like in butterflies, there can be over 100 chromosomes. There's a wide range in the total number of chromosomes and the karyotype across lots of different insects. Sex determination also varies in insects. So there are XY systems, which is known as male heterogametry, which is like what we see in beetles. There is female heterogametry, which is what we see in butterflies, which are ZZZW. There's also haplodiploidy, which is in Hymenoptera. And then there's also kind of some weird stuff going on with paternal genome elimination in some other groups. So there are sex chromosomes in certain groups of insects. And for today, we're going to be talking primarily about beetles, shown here. So one of the interesting observations that's been seen recently is that there seems to be sex chromosome turnover, across different groups in some cases. So in contrast to what we see in something like mammals, where the X chromosome seems to be conserved over long periods of time, and things like Diptera, different chromosomes actually act as the X chromosome or the Y chromosome in different lineages, meaning that these things can change quite a bit over evolutionary time. And the sex chromosomes can be one of the most interesting parts of a genome for a number of different reasons, which I'll detail here in just a second. And how do we actually get to sex chromosomes? There's a general model, which is shown right here, where at one point there can be autosomes. We then get some sort of sex determining region is located on one of the chromosomes, and that gets linked up with a locus that is under some sort of sexually antagonistic selection that can then favor inversions to link those two things together. And then over time, you can get the proliferation of certain types of genes onto the sex chromosome. And then the one chromosome will stop recombining with the other, leading to degeneration to the point to what you see here at the very end, where we might have this degenerated Y chromosome, doesn't have very many genes and tends to be enriched for male-specific genes in a situation like an XY system shown here. Sometimes these heteromorphic sex chromosomes are kind of old chromosomes confused to other autosomes, causing what are known as neosex chromosomes. And I'm going to detail what that looks like here. So imagine we've got this ancestral karyotype shown here. We've got this X and this Y chromosome. And then in this case, maybe the Y chromosome gets lost. The X chromosome is fusing here shown here is like the largest autosome in the genome. This leads to an ancestral X portion of the chromosome, a neo-X portion of the chromosome, and then the homologue of that becomes the neo-Y. So we get this kind of new karyotype through a sex chromosome autosome fusion. Once the neo-X and the neo-Y stop recombining, these things can start to degenerate and differentiate from each other. So one group that has not really been studied in trying to understand kind of large-scale patterns of chromosome conservation and sex chromosomes are beetles. And as many people here know, beetles are a very diverse group, easily over 400,000 described species. And so that's something that's been interesting to me for a long time. And so today I'm going to talk about some of the research that we're doing, looking at sex chromosome evolution and then neo-sex chromosome evolution in specific species. And so, yeah, there's going to be two stories here. One of these looking at kind of broad scale patterns of sex chromosome evolution in beetles, the other looking at neo-sex chromosomes. And these are the papers that are associated with these parts of the talk. So for the first part, I'm going to talk about sex chromosomes broadly in beetles. And I want to just acknowledge kind of up front my postdoc advisor who helped me with some of this research, as well as the undergrad who was doing some of the bioinformatics along with me, and then a large number of people that were also part of some of the genome sequencing. And so the questions here, is the X chromosome conserved like in mammals, or is this something that turns over quite regularly like what we see in flies? Is there any kind of broad patterns of chromosome conservation or evolution in beetles? And then we want to know, does the X chromosome show enrichment or depletion of sex-biased genes like what we see in lots of other systems? And then how quickly do neo-sex chromosomes evolve these types of properties? So I looked at 12 beetle genomes at the time, and shown here are the different species, as well as their different karyotypes and their different types of sex chromosomes. So some of these have neo-sex chromosomes, some of these are X naught, some are XY. And this group, we're talking about species that some of them diverged over 350 million years ago. And so I looked at some published chromosome level assemblies for the species that are shown here in blue. I then also analyzed some published fragmented assemblies, which are shown here in green. And for these species, we generated male and female Illumina data so we could try to identify the sex-linked scaffolds in those assemblies. And then we also did some de novo chromosome level assemblies for a couple of species, which are shown here to kind of fill out some of the phylogeny and try to get better understanding of some specific groups. And I just want to highlight here that we're looking at a couple different species with neosex chromosomes, one here, and one here. But for the purposes of today's talk, we are only going to be talking about these two species here. So we did a genome assembly for Tribolium confusum, which is closely related to Tribolium castaneum, which is kind of the model beetle species. This beetle has a neosex chromosome, and what I'm showing you here is the results of that assembly. This is using nanopore plus high-C and then some Illumina data to polish it. We get near chromosome level assemblies for everything but a few of the chromosomes. We see the blue here is showing you the proportion of repetitive sequences. The portion down here is showing you the gene density across the chromosome, and then the little things down here are stitch points, kind of where we're scaffolding everything together. So we have a really high quality assembly for Tribolium confusum. The genome assembly is around 320 megabases. When we do a synteny comparison between Tribolium confusum and Tribolium castaneum, what we show here is that the X chromosome and castaneum is actually fused to chromosome 2 in Tribolium confusum. So they do indeed have a large neosex chromosome, and we see very clear levels of synteny across the chromosomes, as you might expect by things that are kind of closely related to each other. And there's around 9,000 orthologs that are being shown here with these little lines. The next question we were interested in is just how degenerated is this chromosome 2? And so what we did is we looked at male and female sequencing coverage over the female assembly, and that's what you're seeing here. And the expectation is that if it's equivalent, it should be around 1. And if there is only male coverage, you'd be around 0 here for the female divided by male coverage. And then if we have an excess of female coverage, it should be over here at this red line. And what we see over the entirety of the chromosome is that we have double the coverage in females, suggesting that the neo-Y chromosome has completely degenerated in Tribolium confusum. Turning our attention to another neosex chromosome in Dendroctonus ponderosi, what we see is also quite a bit of clear patterns of synteny across chromosomes, and these two species last shared a common ancestor over 200 million years ago. We also see the X chromosome here is fused to what looks like primarily linkage group 4 in Tribolium castaneum, as well as a little bit of linkage group 2. So we see a clear neosex chromosome in this system. When we do similar types of coverage analyses, we see something that's actually quite different. We see what looks to be a younger neosex chromosome in these beetles. So we have the ancestral X portion of the chromosome here. We then have this large swath of the chromosome where the male reads, so actually the Y chromosome reads, actually map to the X chromosome, suggesting that the neo-Y is not as degenerated. When we look at chromosome conservation in beetles across the phylogeny, what we see is large scales of clear conservation, and we see that there seems to be lots and lots of synteny between Tribolium castaneum as well as the other species that had chromosome level assemblies. And if we draw kind of links between all of the one-to-one orthologs that we can identify across all of the different species, we get this kind of crazy looking thing here. But the main point here that I'm trying to make is that you can see that there seems to be a kind of shared core set of what are called, we like to call them elements. You can think of them as molar elements that are in Drosophila, or they're Nigon elements in nematodes. So there seems to be this kind of conserved chromosome across beetles, and we see this across all of the different species, and we find what we think are around 9 elements and what looks to be X chromosome conservation across the chromosome level assemblies. We then turned our attention to the fragmented assemblies, and using differences in male and female coverage, we identified scaffolds that look to be X-linked in those species. We then identified the one-to-one orthologs that were on those X-linked, putative X-linked scaffolds, and then found where they were in the tribolium confusum or in castaneum assembly. And we see that they all are on the X chromosome and tribolium castaneum, suggesting that the X chromosome is also the same chromosome in all of these other species of beetles. When we turn our attention to the kind of core set of X-linked genes that we can identify across all of these chromosome level assemblies, we then look at kind of where they are located across the chromosome. What we see is this kind of total shuffling up of their location. So although the X chromosome seems to be conserved in beetles, we are getting collinearity across the chromosome is deteriorating over time, and genes are getting shuffled up quite a bit across the chromosome. Looking at gene expression analyses to try to understand if the ancestral X shows an enrichment or depletion of sex-biased genes, what we see is something that has been demonstrated in other species like Tribolium castaneum, and that the ancestral X chromosome seems to be enriched for ovary-biased genes, so genes that show expression in the ovary or heightened expression in the ovary. And it seems to be depleted of genes that show testis bias expression. So this is for the X chromosome, kind of the old X chromosome in Tribolium castaneum. When we turn our attention to Tribolium confusum, which has a neo-sex chromosome, we see that same bias. So we see an ovary bias for the ancestral X, as well as a depletion in testis-specific genes. However, for the neo-X, it looks primarily like a autosome for the most part. We see the same kind of pattern when we turn to Dendroctonus ponderosi, where we see this ovary bias, and we see a depletion of testis bias genes on the ancestral X. However, the neo-X, again, seems to look very similar to autosomes. So it's not showing any kind of sex bias expression in this case. Looking at the kind of conserved set of genes that we find on the ancestral X chromosome, what we see is that these genes are highly expressed in the ovary. So what you're looking at here is gene expression in testes, ovaries, and then the male and the female. So this is gonadectomized males and females, and these are heads of males and females here and here, and then heads of males and females here, with two replicates of females. And so what we see is that for genes on the X chromosome, they're enriched for ovary expression. They seem to be enriched for ovary expression in Tribolium confusum, as well as enriched for ovary expression in Dendroctonus ponderosi. And we see some levels of kind of decreased levels in testis expression, which suggesting that this X chromosome is kind of taken on this potentially kind of feminized role. When we look at the global expression of the X chromosome in males and females in undifferentiated tissues, so basically asking, is there levels of dosage compensation? So because males are only going to have one copy of the X chromosome, whereas females are going to have two, most systems evolve some sort of way to have that gene expression be equivalent in males and females. And what we see when looking at the mountain pine beetle system, looking at the neo-sex chromosome as well as the ancestral X chromosome, which is shown here, we see roughly equivalent levels of male and female expression in the heads, suggesting that both the ancestral X chromosome as well as the neo-X chromosome shows dosage compensation. When we look at Tribolium confusum, we see the same thing. So the ancestral X chromosome shows dosage compensation, as well as the neo-X chromosome shows dosage compensation as well. When we turn our attention to looking at testes and ovary expression, what we see in the mountain pine beetle is this clear pattern where the ancestral X shows kind of this heightened expression in the ovaries and a decreased expression in the testes, and then roughly equivalent expression kind of all over the neo-X. This is kind of looking at global patterns of gene expression across the chromosome. When looking at Tribolium confusum, what we see is kind of overall lower levels of testis expression compared to ovary expression when looking at that chromosome. And just to mention that it is a little bit weird kind of trying to compare testes to ovary because we're dealing with different types of tissues. And so, but there does seem to be some kind of clear differences going on in gonads. And this has been some things that have also been seen in other systems like in butterflies. So in conclusion, we see evidence of chromosome level conservation in beetles across over 350 million years of evolution. We think that this standardized element nomenclature will help aid in future studies so that we're kind of talking about the same chromosomes across systems. The X chromosome appears to be conserved and appears to be enriched for female biased genes. And neosex chromosomes appear to evolve repeatedly, but appear somewhat slow to take on some of these kind of ancestral X properties in some of these systems. And so this is an exciting group to work on to try to understand X chromosome and X chromosome evolution. And with all of the new assemblies that are coming out, we're really excited to dig into even more of them and try to look for some more like exceptions to the rule. But for right now, it looks like the X chromosome seems to be largely conserved across beetles. So I now want to talk just kind of quickly about neosex chromosome evolution and then how it could be linked to speciation. So linking kind of all of these kind of changes that we're seeing with sex chromosome autosome fusions. And this is going to be work that I've been doing in the mountain pine beetle system. And I want to quickly mention some of my collaborators. These have been people that I've been working with for quite some time. And then I want to give a shout out to Scott Gieb, who's been helping with the chromosome level assemblies that we're doing, which are really, really exciting. Okay, so just to kind of quickly rehash, sex chromosomes are special and they are also kind of weird. So, you know, these chromosomes at one point in their history can be autosomes, and then they kind of take on these crazy properties where the Y chromosome becomes degenerated, gets enriched for male-specific genes, and stops recombining with the X chromosome and accumulates lots of repetitive elements. And it's known that the sex chromosomes are important in speciation. This was pointed out a long time ago by JBS Haldane. This is known as Haldane's rule, and this is that the heterogametic sex tend to have problems in crosses and hybridization events. And for several decades now, we've known about this thing called the large X effect, which just shows that the X chromosome tends to play a large role in the evolution of hybrid sterility and hybrid inviability. And this is just from this classic study where they kind of mixed up the chromosomes between two species of Drosophila. And whenever they had a blue X chromosome, the hybrid males were sterile. This is a chromosome from one of the species. We also know that reproductive incompatibilities tend to increase with genetic distance between species. This is also somewhat related to the sex chromosome thing. So as we get increasingly genetically differentiated, we get increasing levels of reproductive isolation, post-zygotic isolation, hybrid sterility, and hybrid inviability between populations and species. So one of the things that we don't know very much about is if neosex chromosomes could play a role in speciation. So we know the old sex chromosomes do, but we don't know much about the neosex chromosomes. We also don't really know how rapid neo-Y chromosomes can differentiate and if this differentiation could lead to reproductive incompatibilities. And so the group I've been working on is to study this is Dendroctonus. These are known as the tree killers. This is a group of very important forest insects. Many of these kill healthy trees. There's around 19 species, which many of them are shown here, and numerous species have neosex chromosomes. And today I'm going to be talking about Dendroctonus ponderosi. This is one of the species I was talking about before. We're going to go into a little bit more depth into this species. This is its karyotype, has 11 autosomes and a neo-XY. So this beetle is a generalist with 5 major pine hosts. It's pretty broadly distributed across Western North America. As I mentioned, it's one of the most significant forest pests. It can disperse over 40 to 50 miles during an outbreak. This beetle has large neosex chromosomes, which were identified back in the 60s. And there's even children's books written about this beetle. So it's a pretty important critter. So some work that I had done way back during my master's and then through my PhD demonstrated that this beetle actually seems to be kind of like a species complex based on patterns of reproductive incompatibilities. And what I showed, and try to summarize this kind of quickly, is that there seems to be some sort of zone or geographic break in the middle of this species distribution, which is kind of shown in gray here. And as you cross populations of increasing distance with respect to this zone, you get increasing levels of hybrid male sterility and hybrid male inviability. So, for example, it's weak and asymmetric hybrid sterility here, and then it's strong symmetric hybrid sterility and also hybrid inviability when we cross beetles from here. And this is a result of, looks like a breakdown in spermatogenesis that occurs in hybrid males, as well as these hybrid males have very long development times and likely would die if they were to occur in nature. So just to kind of quickly point out what some of these methods that we use look like, this is how we go about crossing some of the beetles to try to get some of these results. So I've done some population genomics analyses in the past to try to understand the population structure for this beetle. So this was whole genome sequencing data around 31 individuals. There was a reference genome. This was the second beetle to have a reference genome published for it. So we were able to take advantage of that. And using over 1 million SNPs partitioned across autosomes and the sex chromosomes, what I was able to show is that if we look at the autosomes, this looks kind of like one species. It shows this isolation by distance, kind of gene flow or genetic pattern kind of around the Great Basin Desert. However, if we look at SNPs that are from the neo-sex chromosomes, and particularly the neo-Y chromosome, we see what looks to be 3 very distinct Y chromosomes, and they seem to kind of correspond to where this break occurs within the species, suggesting that maybe these sex chromosomes are playing some sort of a role in the incompatibilities. Digging into genome coverage over the genome, and during some of these analyses, I was able to find that there are actually contigs in the assembly where we were not getting neo-Y sequencing coverage in Western beetles or in Southeast beetles. And so we were very curious about the fact that it looked like the neo-Y chromosome is kind of degenerating in different ways in different populations, and again, seemed to kind of correspond to where we were seeing breaks in the reproductive compatibilities. Within those regions that seem to be missing from the Y chromosome in certain populations, we were able to actually find genes, and these genes are expressed heavily in the testis, and this gene is even known to play a role in fertility in, say, Drosophila and in other organisms. So some of these Y chromosome deletions may have genes that are actually important to the species, and there seems to be kind of a mixture of what genes are on some of these Y chromosomes in different beetle populations. So some very interesting things are going on with the genome, but this was a study that was done a number of years ago with a fragmented assembly. And so we're very excited to kind of jump in and use some new techniques to really understand what's going on with the sex chromosomes in the mountain pine beetle. And so I'm going to talk a little bit about some of the progress that we've been making to understand the neo-sex chromosomes in the mountain pine beetle and the link to some of these hybrid male problems that we observe when some of these crosses. And what I'm very interested in exploring is what is the extent of this neo-X and neo-Y differentiation. So as I mentioned, as soon as these things stop recombining with each other, the neo-Y can start differentiating from the neo-X. It can start degenerating as it can pick up deleterious variation that it can't purge. And then I'm also very interested in trying to understand neo-Y structural variation among the population. So looking at multiple Y chromosomes to understand how these things are diverging from each other. And so what I'm going to talk about here today is going to be some assemblies and some analyses that we've been doing between populations from Oregon, Idaho, and Nevada, which represent the 3 kind of Y chromosome types in the mountain pine beetle. So we have been doing, like many people, PacBio Hi-Fi and Hi-C sequencing and doing single males and single females from these populations to get very high quality assemblies of the autosomes and the sex chromosomes for comparative purposes. And so what I'm showing you here is a Hi-C contact map. You can see the 11 autosomes, which are shown here. We then have the X chromosome, which is shown here. And then we have the Y chromosome. And I just want to point out here, so the blue boxes are the chromosomes, and the green here, the scaffold, sorry. And then the green is showing you the contigs. And what you can clearly see here is that we're assembling the X and the Y in just a few contigs, which is pretty awesome and quite exciting. And then all of the autosomes are being assembled in a single contig. So we have these for males as well as females from all of these populations, although I'm only showing you the male example from Idaho shown here. So for those who are interested in these types of things, Busco statistics and stuff, here are some of the numbers. So these are what the chromosome lengths look like. So the X chromosome is around 70 megabases. The Y chromosome is over 50 megabases. And then all of the autosomes are around 20 megabases or smaller. So you can see the Y chromosome is actually quite large, as is the X chromosome. These are bigger than many of the autosomes. Or most of the, or sorry, all the autosomes, but even, you know, multiple autosomes if they were to be stuck together. The Idaho, Nevada, and Oregon Busco results are shown right here. You can see that the assembly completeness is quite high. You'll see that there's a number of duplicated here, and that's because we have the X and the Y both in the assembly, so we have duplicated across those two chromosomes. I'll talk about that here in a second. But you can see these assemblies are quite good. We are getting the X and the Y and everything else, which is quite exciting. So one of the things we were very interested in is trying to look at collinearity across genomes for the X and the autosomes for all of these different populations, Oregon, Idaho, Nevada. I'm not going to show them all here today, but I'm just going to quickly show you what Nevada and Idaho looks like. So this one and this one. And what we would expect if these things are perfectly collinear is that we should just get this line kind of right on the diagonal as we're comparing these two genome assemblies. And that's pretty much exactly what we see. So the X chromosome looks to be collinear with the other X chromosome. We do pick up what looks like a small inversion on chromosome 2, but by and large, the genomes, when comparing X chromosomes and autosomes across all of these three different populations, look very, very similar and are very collinear. So one of the things that we're very interested in is what actually then fused to create the neo-XY in this system. As I mentioned, those chromosomes are actually quite large. And just as kind of a quick refresher, what we're talking about here is a situation where in the ancestor, and the ancestral X chromosome would have fused onto this neo-X. What autosome was it that it actually fused to? And so what we've done is we've looked at one other chromosome level assembly that's been published within the group, Valens here, which has 13 autosomes. And when we compare the Buscos between these two genomes, what we see is that the X chromosome and Dendroctonus ponderosi seems to be a composite of the ancestral X chromosome plus what is numbered as 1, 11, and 4 in their assembly. And then all of the other chromosomes pretty much seem to be about a 1-to-1 kind of match. So suggesting that in ponderosi, we might have almost up to 3 different autosomes have actually fused on to create this large X chromosome that's a combination of an old X and a new X. So the next question is, how differentiated is the neo-X from the neo-Y chromosome? And how long ago have these things stopped recombining, or have they stopped recombining at all? Do they still just look like autosomes? What we find is that there are numerous structural changes between the neo-X and the neo-Y. So here is showing you the neo-X on this axis and then the neo-Y on this axis. And you can see that this is totally jarbled up. So it looks like there is massive structural differences between the neo-Y and the neo-X, suggesting that these things are highly differentiated from each other. When looking at the neo-Y, we see that it's differentiating and accumulating repeats, but still has some functioning genes. So here is showing you the X chromosome, and here is showing you the Y chromosome. The gray here is showing you kind of repeat density across the chromosome. There is actually an increase in the amount of repeats that we see on the Y and compared to the X. Then what I'm showing you here below are the genes with an X chromosome and a Y chromosome copy. So these are genes that are still conserved across the neo-X and the neo-Y. And then in black here are just the genes where there's only an X copy. So meaning that we can find some genes that are still shared between the neo-X and the neo-Y, but then there's also a bunch of genes where we cannot find the Y version. So how differentiated are the neo-Ys from each other across these different populations? So I'm just going to show you Nevada compared to Idaho here, but there are numerous structural changes between Idaho and Nevada neo-Y. So you can see massive numbers of inversions, large ones and small ones that shuffle up these genomes. And when we've looked at the BUSCOs, we're still working on the annotations for these neo-Y chromosomes, as they're kind of challenging because genes are degenerating on them. But when we look for just the BUSCOs across these chromosomes, compare where they are across these different neo-Ys, we see that there is extensive shuffling of all of these genes across these chromosomes. So they're just linked here by color. We see that there's around 16 BUSCOs missing from the Nevada Y chromosome that we find on the other ones. We see there's 17 genes missing from the Idaho Y chromosome and around 20 genes that seem to be missing from the Oregon Y. So we have these kind of unique sets of genes that seem to be missing from the different Y chromosomes from the different populations. So in conclusion to the second part, the neo-Y appears to be degenerating quite rapidly in the mountain pine beetle. The fact that we seem to see different sets of genes degenerating or being lost from different Y chromosomes suggests that these kind of processes could be playing a role in hybrid sterility. And these really is an exciting thing to think about, that these sex chromosome fusions could really be setting the stage for these kind of rapid genomic changes to be occurring in isolated populations that then could result in new species forming. And then to just zoom back out and think a little bit about sex chromosomes more generally, we're actually finding more neo-sex chromosomes in species because of all of these new long read sequencing assemblies and really high quality assemblies. So now there's neo-sex chromosomes that have been found in parrots, where we have this chromosome is fused onto the Z. We also know about neo-sex chromosomes in butterflies. And I think as we get more and more assemblies, we're just going to find more and more of these neo-sex chromosomes and maybe find that they are playing kind of important role in speciation in certain systems. And so with that, quick acknowledgements for a lot of other people who have helped that I didn't already point out earlier, maybe helped with beetle collection or with some analyses or part of the lab and had seen and helped with certain parts of these talks or certain wet lab techniques. And with that, I'll open it up to questions. Thanks.
Thank you, Ryan. That was an excellent talk. We do have quite a few questions coming in. So I'll go ahead and read those aloud and then you can go ahead and answer them.
Sounds good.
So let's see. Erica is asking, are there any general patterns in the elements that show broad synteny compared to other scaffolds that pop up and disappear? For example, maybe transposable element content?
Yeah, so that's an interesting question. So you may have noticed that there's one of those chromosomes or elements that it kind of gets left off of the element naming because I think it's like one of the smaller chromosomes in Tribolium castaneum. We just can't really find it in any other genome. It's kind of something that seems to only really be in that one species. And there are some little bits that do seem to kind of pop up and then go away in other assemblies. And I just don't know the answer to that yet. I think we need to have a little bit better kind of like ancestral state reconstruction of the chromosomes across Coleoptera to get a better sense as to what little bits maybe do kind of break off or have their own weird kind of trajectory within the group. How that relates to transposable elements, I'm not quite sure. But yeah, there's definitely more to explore here. So
cool. Cool. Okay. Let's see. Is the gene order reshuffling significantly higher in the X chromosome in comparison to autosomes? And if so, what are some possible explanations for that?
yeah, so that's also a very interesting question, something that I haven't explored quantitatively. I've seen that similar type of a pattern when looking at Drosophila stuff as well. So I've done a lot of kind of comparative genomics in that group, kind of looking at those transitions. And by eye, I tend to see what looks like lots more of inversions, even between kind of closely related species on X chromosomes versus autosomes. Some of that's thought to be maybe due to the lower effective population size. Things can maybe meiotic drive. There's lots of different reasons why we could explain kind of weird things maybe happening on X chromosomes in contrast to the autosomes. But I haven't really done any kind of quantitative analysis of that. And so that's waiting, but very interesting question, something that should be explored.
I was a little curious about, or spiked my interest, the fusion of multiple autosomes with an X to make that giant X chromosome. Are there known mechanisms that cause the fusion of chromosomes, or hypotheses about why that would happen?
It's a little unclear. Maybe just happens by chance. If you have two telecentric chromosomes, it's kind of easy to imagine how they could just kind of get stuck together. And in like Drosophila, where people have kind of studied this quite a bit more, we do see just kind of aberrant chromosome morphologies pop up in certain lines and stuff like that. So as to like a mechanism, people have proposed, again, some of these kind of meiotic drive type mechanisms, but it's unclear what kind of role they might actually have and if that's real. But the idea being that maybe if you have this fused chromosome with a really large centromere, that it may kind of mess with meiosis and have some selective advantage. But that really hasn't been shown or proven in any system as far as I know, at least in terms of chromosome. So yeah, it's with that story with the multiple autosomes sticking to each other. It's also a little unclear if those were maybe autosomal fusions prior to the X, or if that's like the X sequentially stuck pieces of autosomes on it over time, and we're trying to figure that out right.
Okay. We have a Weevil genome that will be out fairly soon. That may be a good addition to your data set.
Yeah, exciting to see it. Yeah.
Okay, David Heckel is asking, is the X chromosome of beetles related to X chromosomes? He says in locusts, but maybe other insects. Maybe he's asking if they're conserved across all insects.
Yeah, so when I started on the beetle thing, I just kind of stuck to beetles, and then over the last couple of years, there's been a lot of papers that have come out suggesting that some of these sex chromosomes are maybe conserved further back, across like arthropods or things like that. Or there's even like really deep conservation of chromosomes across metazoans and stuff. And I just never really kind of jumped further back out of Coleoptera. So that's something that would be interesting to explore, but I haven't really looked any deeper into the tree past kind of Coleoptera.
Very cool. Okay, one last question. Where do you expect to find candidate HMS genes involved in the ceratogenesis abnormalities in hybrids? On the Neo-X or on the Neo-Y? What approaches would you use to identify those candidates?
Yeah, we're working on that right now. I'm not going to give it away. We have ideas as to where they're going to be. We think that the Neo-Y seems to be degenerating in different ways in different populations. As if it's a gene that deleted on the Y, and then the X has to figure out how to handle the fact that that Y gene is missing. It's a little bit unclear what happens there, but we imagine that basically what is causing incompatibilities in the mountain pine beetles, we have mixed up kind of X's and Y chromosomes that have different gene contents, and you can imagine that could lead to some sort of problems in hybrids. But as to what specific genes, what chromosomes are on, which ones are doing what, that's what we're working on right now. So stay tuned. We hope to have some answers.
Very cool. Sounds great. All right, I feel like that was a good one to end it on for now. And like I said previously, if you have questions that come up later, you can type them in and we can get to them at the very end, potentially.
Cool.
But with that, though, let's move to our last speaker of the day. Our speaker is Michelle Jonika. She also has interests in sex chromosome evolution and the evolution of other genomic features as well. She did her PhD here at Texas A&M University, and she has stuck around to do her PhD with Heath Blackman, who was a speaker of this symposium a couple years ago. So with that, let's start with Michelle's talk.
My name is Michelle Jonika, and I am a postdoc researcher in Dr. Heath Blackman's lab in the Department of Biology at Texas A&M University. And today I'm very excited to talk to you about the coevolution of binary traits in genome structure in insects. The study of chromosomes has a very rich history, with the discovery of chromosomes in 1882 by Walter Fleming to the discovery of sex chromosomes by Nettie Stevens in 1906. And our lab even hosts a website, karyotype.org, which contains more than 30,000 publicly available karyotype records across the tree of life. Yet, despite all of this information and this very early discovery of chromosomes, we can see from people such as Maynard Smith in 1998 that we have a very poor understanding of the causes of karyotype evolution despite many careful investigations, and the general significance of karyotype evolution is obscure. And even more recently, from Matthew Rockman in 2007, we see that the roles of natural selection, drift, and mutation in shaping the evolution of karyotypes has been largely refractory to broad generalizations. And this has always been something that really drives our research into chromosome number evolution in the lab, is wanting to know this seemingly basic yet very important point of what impacts or causes chromosome number evolution. And so why do we research chromosome number evolution? The example pictured to the left here is why. So these two ants, which are in the same genus and are actually even sister species, have evolved these fundamentally different chromosome numbers. So we have the one species on the left with a diploid chromosome number of 2, and the other species on the right with a diploid chromosome number of 42. And this is just one example here, but we also see this pattern throughout large clades across the tree of life. And we specifically see this in insects. So in this plot pictured here, we have more than 16,000 chromosome number observations. Along the vertical axis, we have a phylogeny of insect orders, and then along the horizontal axis is the haploid autosome count. And within each bin of haploid autosome count is a color that's based on the number of observations within that order for the given autosome count. And so warmer colors are going to indicate a higher number of observations, while cooler colors are going to indicate a lower number of observations. And I'm going to have us focus in on 3 very specific orders: Diptera, Lepidoptera, and Coleoptera. And these are 3 orders that have a very similar number of samples for the clades and similar ages. However, Diptera and Coleoptera have a very tight range in chromosome number. So we see from 2 to 13 in Diptera and from 2 to 36 in Coleoptera, while Lepidoptera has a very wide range in chromosome number, from 5 all the way to more than 200. And we observe this very striking heterogeneity in the range of chromosome numbers that are present among clades. And this tells us that there must be a great heterogeneity in rates of chromosome number evolution among lineages. And despite many recent developments in comparative methods, most of this heterogeneity is still poorly understood. And so today in my talk, I'm going to give an overview of our lab's chromosome number evolution model, which can study the impact of traits on the evolution of chromosome number across large clades and millions of years of evolution. And 2 such things that I'm going to discuss today that involve that model are the trait of centromere type, as well as the fitness effects of changes in chromosome number. And then finally, I'm going to end using a simpler model of chromosome evolution to discuss the rate of chromosome evolution broadly across the insect tree of life and the implications of these results. And so up first, I'm going to introduce Chrome+, which is a model for chromosome number evolution. And this model is going to underlie all of the research that I'm going to be discussing with you today. My PI, Dr. Heath Blackman, created a biologically realistic model to estimate rates of chromosome number evolution called Chrome+. And the inputs for this model in its simplest form are just a phylogeny and chromosome numbers. And so at an instance in time, a lineage will have I chromosomes. And this model contains 3 mechanisms for changes in chromosome number. We could have a fusion of 2 chromosomes, I-1. We could have a fission of a chromosome, I+1, or we could have a whole genome duplication, 2I. But we can also add more information to this model. We could also give information on a binary trait for a given phylogeny and the corresponding chromosome numbers. And so we've done that here, where we have state 1, which is in purple, and we have state 2, which is in pink. And this is actually going to add 2 additional parameters into the model, which will describe the transition to and from each binary state. So we can have a transition from state 1 to state 2, or a transition from state 2 to state 1. So taking this more complex model in, at an instance in time, a lineage will have I chromosomes and either be in binary state 1 or binary state 2. And we can have 8 total parameters that are calculated in this more complex model. So again, we have those 3 mechanisms for changes in chromosome number: fission, fusion, and polyploidy. And each of these is estimated separately in each binary state. So we can have fission, fusion, and polyploidy in state 1, and fission, fusion, and polyploidy in state 2. And so this leads to 6 chromosomal rate parameters. And then we also have the final 2 parameters, which will describe the transition to and from each binary state. So the transition from state 1 to state 2, and the transition from state 2 to state 1. And so for ease of interpretation, we present results from this model as a mean rate difference across our posterior distribution of trees for a given clade. And that mean rate difference is pictured under the model on the left-hand side of this slide. And so in this example, we have the mean rate difference in fusions, where we calculate delta R of fusions equals the fusions in state 1 minus the fusions in state 2 to estimate the magnitude of each parameter, or in this case, the fusion parameter. And in addition, we compare the 95% credible interval for each of our parameters to 0. And so if the entire 95% credible interval of our delta R fusion, which we're using this example, is negative, we interpret that as support for a higher rate of chromosome number evolution in state 2. If the entire 95% credible interval overlaps 0, we would conclude that there is not support for a significant rate difference between state 1 and state 2. And finally, if the entire 95% credible interval of our delta R fusion is positive, we interpret it as support for a higher rate of chromosome number evolution in state 1. And this model of chromosome number evolution is publicly available as an R package, Chrome Plus. And this comparative method will allow us to identify co-evolving traits that lead to heterogeneity and rates of chromosome number across the insect tree of life. And so now I'm going to apply that into several research projects that I've been a part of within the lab. And the first thing that I'm going to talk about is the trait of centromere type in chromosome number evolution. And so in this plot, I again have haploid chromosome number along that horizontal axis, and then each row is an insect order. And Lepidoptera is conveniently highlighted in that red box. And again, you'll remember that the range of chromosome numbers in Lepidoptera is from 5 to all the way more than 200. And pictured in this plot, I really just want you to take in that when we look at this order in relation to all of the other orders in insects, you can see that it's taking in the full span of chromosome numbers that we observe in insects. And this idea had resurfaced recently in a Nature paper that I read a few years ago in a journal club that claimed that holocentricity was the explanation for extreme chromosome number in some lepidopterans. And this is actually an old idea that's been driven by the kind of data distribution that you see here. And so people very early on began to say that this is due to that order having holocentric chromosomes. But we decided that we really wanted to dig into this more because no one had ever really tested that. And so when you actually look at chromosomes and the type of centromeres that they can have, so monocentric versus holocentric chromosomes, it kind of makes sense, this thought. And so when we're looking at it here, monocentric chromosomes, which are pictured on the left, are going to have a single centromere that's used for spindle fibers to attach during meiosis, while holocentric chromosomes, which are on the right-hand side, have a centromere that's diffused across the entire length of the chromosome. And so those spindle fibers are going to attach at each of those centromeres during meiosis, and then the meiotic products will segregate into their appropriate gametes at the end of meiosis. And so that's great, but what happens if we have something like a fission event that occurs? Now we'll walk through that same process again. So again, those spindle fibers are going to attach during meiosis, but now in the gametic products that are produced, we can see that how each of these centromere types resolves this fission event is very different. And so the monocentric chromosome, which is again pictured on the left, will have some gametes that do have appropriate genomic content, while others that are either missing or have extra genomic content. Whereas that holocentric chromosome that's on the right produces gametes with appropriate genomic content. And for this reason, it would make sense that holocentric chromosomes may be more tolerant to changes in chromosome number. But we wanted to actually apply some data to this and look further into this longstanding thought within the field. And so pictured here, I have a general level phylogeny with orders with holocentric chromosomes painted in gray and orders with monocentric chromosomes painted in black. And we have 3 holocentric orders that are within this phylogeny, as well as 7 monocentric orders. And we also additionally, on this plot, have the chromosome number for each of these genera, where the tips on the plot are going to be labeled or colored, based on the order for each genera, and the height of each tip is going to correspond to that genera's haploid chromosome number. And so what we want to know from this data is whether gray clades, or those orders with holocentric chromosomes, are going to have higher rates of chromosome number evolution than the black clades, or those monocentric clades. And our model of chromosome number evolution that we're going to be using with our binary trait is pictured to the right of this plot. And our delta R statistic that we're going to use is monocentric minus holocentric for each rate. And therefore, if the rates are positive, we expect that monocentric clades are evolving faster, while if the rates are negative, we expect that holocentric rates are evolving faster. So what happens when we dig into the data for this? So this figure on the left-hand side shows the highest posterior density of the delta R statistic along the horizontal axis, where the credible interval of each rate, fission, fusion, and polyploidy, in pink, green, and purple respectively, is located under each density curve. And I have the model that we ran as a reminder pictured above the plot, as well as that delta R rate difference equation. And we can see that all three credible intervals for fission, fusion, and polyploidy overlap 0, indicating that we can't say that either monocentric or holocentric clades are evolving faster rates of chromosome number evolution. Now, there's only mild support for polyploidy across the insect tree of life, and so we wanted to be sure that our rate estimates for polyploidy weren't driving the results that we were seeing. So we also additionally ran this model without polyploidy to see if our results changed. And when we run this model, we again find that both fission and fusion have credible intervals that are overlapping 0. Another test that we ran in this study is we wanted to run our simple model of chromosome evolution. So without a binary trait, looking at fission, fusion, and polyploidy rates for each order. And that model is pictured above the plot here. And along the horizontal axis in this plot, we have the 3 rate parameters, fission, fusion, and polyploidy. And within each of those rate parameters are the 3 holocentric orders first, and then the 7 monocentric orders to the right of those. And we can begin to notice some patterns in this data, such as Lepidoptera, which is that first bar or order that you're going to see in each rate parameter that's in pink or a fuchsia color. And we see that Lepidoptera has very high rates of fission, fusion, and polyploidy. However, the other 2 holocentric orders, Hemiptera and Odonata, which are directly to the right of Lepidoptera, do not have these high rates. And if we look at the 7 monocentric orders that are to the right of these 3 holocentric orders, we notice that some of these orders also have higher or even equally as high rates as Lepidoptera. And so this was really interesting. Again, we wanted to test this without polyploidy in the model as well, but similar to the last analysis, we do still see similar patterns when we run the model both with and without polyploidy. And so therefore, taking all of this data together, when we come back to our question discussing whether holocentricity is responsible for the extreme chromosome number variation that's observed in Lepidoptera, we find that holocentric clades do not generally have higher rates of chromosome evolution. Therefore, we don't believe that holocentricity alone is responsible for the extreme chromosome number variation that's observed in this clade. And so that was a really interesting study. And now I'm going to switch gears into a more recent research project that we've done. It was actually just published this January. And for this project, we wanted to look at the fitness effects of changes in chromosome number. And so in this panel of figures that I'm about to show you, the horizontal axis is going to be the population size class, so small or large, and then the vertical axis is going to be the rate of chromosome number evolution. And so in this first panel that I have pictured here, if changes in chromosome number evolution were neutral, then we can think that the rate of chromosome number evolution would be similar in populations with both large and small effective population size. If changes in chromosome number were beneficial, which is shown in this middle panel, then we can think that the rate of chromosome number evolution would be higher in populations with large effective population size with respect to populations with small effective population size, because selection is more efficient in populations with large effective population size. And finally, if the changes in chromosome number were deleterious, which is shown in the last panel, then we can think that the rate of chromosome number evolution would be lower in populations with large effective population size with respect to populations with small effective population size, because again, selection would be less efficient in populations with small effective population sizes. And so we believe that the fitness effects of changes in chromosome number are deleterious. That's going to be our hypothesis leading into this study and then the following research. And we'll be using several criteria as proxies for effective population size, because effective population size is actually quite a difficult thing to measure and is not really known for many of the species that we are working with. Therefore, we can use traits that are associated with lower effective population size and traits that are associated with higher effective population sizes as proxies for what we would like to use in our study. And an example of this is specialists are typically associated with lower effective population size, while generalists are typically associated with higher effective population size. Animals that are flightless are typically associated with lower effective population size, while animals with flight are typically associated with higher effective population size. And the same can be said for inbreeding versus outbreeding and island versus continental species. And so we can use these traits as proxies for effective population size to look at the fitness effects of changes in chromosome number. And so we did this in the beetles. And so the first thing that we investigated is the rate of chromosome number evolution in Carabidae, the ground beetles, where a portion of the clade is wingless while others are winged. And so in our analysis of Carabidae, we fit 2 models, both with and without polyploidy. And in each plot, each curve represents the posterior distribution of rate difference, where the rate difference can be either fission, fusion, or polyploidy, indicated by the color of the fill, purple, green, or yellow, respectively. And positive values are going to indicate higher rates of chromosome evolution in wingless lineages, while negative values are going to indicate higher rates of chromosome evolution in winged lineages. And values overlapping 0 indicate neither wingless or winged exhibit differing rates of chromosome number evolution. And again, below each curve is going to be that bar representing each statistic's 95% confidence interval. On the far left of this slide is the model without polyploidy, so it has just fission and fusion. And when we fit this model, we find that the rate of fission is higher in wingless lineages, but the rate of fusion was similar in all lineages. And then on the right is the model that included fission, fusion, and polyploidy. And when we fit this model, only the rate of whole genome duplication was higher in wingless lineages than in winged lineages. And we additionally looked at 2 clades, polyphaga and adiphaga, and their distribution of traits that were likely to impact effective population size. And traits were scored based only on species that were included in the analysis and when a genera had more than 15 species. And additionally, the majority state for each genera is the state that was assigned. And so expected effective population size was categorized by the number of effective population size impacting traits in a clade. And so these were things such as wingless, strictly inbreeding, monophagous, and restricted to islands. And so an index was assigned to each of these genera, and then a category of high, medium, or low effective population size were assigned to the clade based on the index that was given. And so these categories of high, medium, and low effective population size were used in the following analysis, where we looked at polyphaga and adiphaga and the difference between the low effective population size clades and the medium and high effective population size clades. And the results from this are actually pretty striking. So for each of the genera that we have pictured here, we plot 10,000 samples from the posterior distribution of mean rate estimates from each generation of the MC that comes from our model. And the mean of the posterior distribution is indicated with a black circle, and the credible interval is indicated with a vertical black line. And rates for polyphaga are plotted on the scale to the left, while rates for adiphaga are plotted on the scale to the right. And the dashed line is going to separate those clades plotted against each of those different axes. And the color of points indicates the effective population size class, low, medium, or high. And in polyphaga, which is the group that is located on the left of this plot, the mean rate of fusions in that low effective population size clades ranges from 0.05 to 0.11. And in contrast, the medium and high effective population size clades mean rates were all below 0.025. And then in adiphaga, which is pictured on the right of this plot, only one clade is classified as low effective population size, Calathis. However, Calathis exhibits strikingly high rates compared to the other 4 clades. So all rates for Calathis were more than 10 times higher than the average of the other adiphaga clades that had medium and high effective population sizes. And so with these two analyses taken together, we find unique insight into understanding the distribution of fitness effects of mutations across the tree of life. And we suspect that changes in chromosome number are likely deleterious, given the observations of high rates of chromosome number evolution in clades with small population size. And so we conclude that the distribution of fitness effects of mutations in a trait are key to understanding how a trait will evolve. And our research really aims to better understand the distribution of fitness effects of mutations that have fixed and produced extant diversity in chromosome number. And so we believe that changes in chromosome number are likely deleterious, leading to high rates of chromosome number evolution in species or clades with small effective population size. And finally, I want to end on a research project where we looked at a simple model of chromosome evolution across the entire insect tree of life. And we didn't include a binary trait. We just wanted to investigate the rates of chromosome number evolution broadly across the insect tree of life. And I'm only going to touch a little bit on it, but I think it's pretty eye-opening for insects to view this data all together. And so plotted on this slide is the rates of chromosome number evolution in insect orders. And in each plot, we show the distribution of the estimated rates for each order of insects included in the study. And on the vertical axis in each order of insects included in our study, and then on the horizontal axis are going to be rates of chromosome evolution plotted on a log scale, but unlogged values are going to be shown for ease of interpretation. And so the credible interval for each statistic is shown as a black line, while the mean is indicated with a black circle. And then additionally above each plot, again, I have that model that was run for each for ease of interpretation. And so that plot on the left is going to be the simple model with fissions and fusions only, no polyploidy, while the model on the right is the more complex model that includes polyploidy. And the insect clades are actually ordered by mean rate and chromosome evolution. And so Odonata has some of the lowest rates, while Lepidoptera has some of the highest rates. And I know there's a lot of data present on this slide between these two plots, but the main takeaway that I want to emphasize here is that we observe fundamentally different rates and patterns of chromosome number evolution in these insect orders. And we specifically see that orders such as Lepidoptera and Diptera, which are those bottom two orders in this plot, exhibit quite high rates of chromosome number evolution, while clades such as Odonata, which is the top order in this plot, have quite low rates of chromosome number evolution. And one challenge that we actually have when we compare across clades is a scaling of rate estimates caused by errors in the depth of phylogenies. So for instance, if a phylogeny is stretched to twice its true depth, rate estimates will be 1/2 of the true value. And so for this reason, we also evaluated the normalized rates ratio across each of these orders. And so we calculated this as the rate of fusion divided by the sum of the rate of fusion and fission. And the normalized rates ratio is not impacted by tree depth. And this allows for comparisons independent of errors in tree depth. And so in this plot, we have pictured on the horizontal axis that normalized rates ratio, where again, the black line indicates the credible interval for the rates ratio, and the mean is indicated by the larger black circle. And then each row of the plot represents an order with a number after the order indicating the number of tips included in the analysis. And again, we see in this normalized ratio for these rates that we observe very fundamentally different rates and patterns of chromosome number evolution in these insect orders. And so here it becomes very easy to see that orders such as Diptera, Orthoptera, and Odonata show these bias towards more fusions than fissions, as their rates ratios are greater than 0.5. And so this helps us to tease apart some of that variation that we're seeing when we observe it in this manner. And so this study really illustrates that both the tempo and mode of chromosome evolution is strikingly variable across insect orders. Specifically, what I was able to talk about with regard to tempo is that we find that some orders have rates that are considerably higher than rates in other insect orders, orders such as Lepidoptera and Diptera. And this suggests that in these clades in particular, mechanisms of speciation that are dependent on or accelerated in the presence of structural changes in the genome may be more important. And in contrast, we find that other orders, such as Odonata, appear to have genomes that are structurally more stable. And so this suggests that in this clade, speciation under models that depend on structural changes will be less frequent. And I didn't really get to touch on it much here in this presentation, but another important finding of this paper is that the approaches that we use provide important insights with regard to potential future sequencing projects. And so, for instance, our analysis in Diptera really point to the need for more whole genome sequencing in clades such as Tabanidae, Stratiomyidae, and Bombylidae to understand how species in these groups are able to form new chromosomes, while species in other clades appear unable to form new chromosomes. And so with that, that wraps up the research that I'm going to be able to show you today. And if any of y'all are more interested in reading more about some of the amazing additional analyses that I was not able to present today, all three of the studies that I talked about today are published. And so the first study that looks at chromosome type is in PLOS Genetics. The next study that looked at the fitness effects of chromosome evolution is in the Journal of Heredity. And then lastly, where we looked at broadly the tempo and mode of genome structure evolution in insects, that article is out in Genes. And so I'd love to answer questions later on if you have any about these projects, or if you'd like to read more, I invite you to look at these papers. And with that, I have been really fortunate to have a very wonderful many years in Dr. Blackman's lab. And so I've had the opportunity to work with a lot of different lab members and get to collaborate with a lot of different people. And none of this research would be possible without many of the amazing lab members and collaborators that I've had the opportunity to work with. And so I'd like to thank all of them, as well as the funding sources that I've had available to me during my PhD and into my postdoc. And so I've been fortunate to have funding support me from the NIH, from the Institute of Data Science at Texas A&M, as well as from the biology department and from my graduate program, the interdisciplinary graduate program in genetics and genomics. And with that, I want to thank all of you so much for getting the opportunity to be able to talk to you today. And I am just very grateful and fortunate to have been asked to be able to talk to you about research that's super important to me and has been an integral part of my PhD and postdoc research.
All right, thank you, Michelle. We have some great questions coming in. Let's see. Oh, Kirsten also turned on her camera. Do you want to go ahead and ask your question first, Kirsten? Oh, that was an accident.
I was turning it on just to turn it on. Sorry.
Oh, okay, fine. I thought you had a question. All right, here we go. So is there any evidence that the general principles you found apply to other major evolutionary groups other than insects? For example, are these questions being asked for animals and plants?
That's a great question. So yes, we are applying this to other major evolutionary groups. So because the scope of this presentation is insect focused, I kept all of the data presented here to insects. But actually one of my dissertation projects involved applying this to carnivores. And we've applied this to other groups as well that are currently being worked on in the lab. And so this is something that's really important to us to look at across the tree of life, not just in insects. But we also are a beetle lab, so y'all are hearing a lot about beetles today. And so we do have a large focus in insects. And so we had quite a few projects where we did specifically focus on insects and the traits that we thought might impact this in insects. But there is more out there in animals. And then there's also other labs that are working on this in plants. And so I think there was a recent paper that came out last year, or maybe at the end of the year before that, that actually looked at this in plants. And so it's being pretty well covered across the tree of life.
And those same patterns still exist. Is that right?
Yeah.
Cool. Very cool. Okay, this is kind of a long one, so hopefully I can read this right. All right, so it's great to see a quantitative test of the idea that Lepidoptera have such chromosomal diversity because of holocentric chromosomes. You conclusively rejected that idea. But why are there two types of Lepidopteran clades? One maintaining the modal number n equals 31 pretty closely, others that show the crazy pattern of fragmentation. Can you use your model to compare these two groups?
Yeah, so as long as you're looking at a group that has probably an appropriate number of observations. So nothing too small. We usually try to have at least like 10 to 15 in whatever group we're looking at to make sure that it's not a low sampling size that's going to be impacting what we're looking at. So as long as the groups you're looking at have an appropriate number of data that can be utilized, then I would say this is something that our model could be used for. And so you could either use it just with chromosome data, or if you think that there may be a trait in these two groups that might be impacting this potential fragmentation that you're seeing, you could also apply that in as well and test whether that trait might be impacting the differences that you're seeing. But I guess another good thing to think about too is phylogenies are probably what limit us the most in what we're applying. So having a good phylogeny for whatever group you're looking at. Chromosome number data is a little more available, probably for doing that reduction in data to make sure you're having all that overlapping data for what you're looking at. So that's also something to consider, as well as the availability of that data in those groups as well.
Very cool. Okay. Thank you. Let's see. Another question here. You found variation between orders. Do you think that certain families or genus really can drive some of these differences?
Yeah, so that's actually something that's currently being worked on to get out in the lab. And so we did look at these on a very broad level with a larger overview, but we also wanted to tease apart some of this variation that is in some of these smaller groups as well. So maybe looking at in just one order, maybe what are some specific traits that maybe specific genera within that order have. And so that is something that's important to us, but we started big and now we're going to try to get into some of those more finer details to understanding some of these unique clades that are present within insects and other groups across the tree of life.
Very good, thank you. I see Ryan put his camera up and I was actually trying to link you all's data in my mind while you were speaking. Go ahead, Ryan, and ask your question. I'm sure it'll be much more put together than mine.
No, it's not, but I'm very familiar with their lab and their papers, and Jonika's papers are all great, or Michelle's papers. And one of the things that I was wondering about is in those models, it's kind of treating it like each chromosome is kind of the same thing. But in those Lepidoptera, would have the crazy chromosome numbers. A lot of the crazy chromosome numbers are kind of driven by microchromosomes, right? And it's kind of unclear if there's even genes on there, or how many genes are on there, and what they're even doing. So I was kind of wondering if there's any effort, or if it's even possible, to kind of try to take into account that like some fusions are maybe going to be more challenging than other fusions, given the fact that like big chromosomes are going to have lots of genes. It potentially could be more deleterious or detrimental if you had a fusion between those chromosomes, versus like if 3 or 5 little micro chromosomes get stuck together, like, does it matter? Just kind of wondering about that.
Yeah, that's a really great question. So currently our model does not take that into account, but that is an important point. We would be more likely to see that in some chromosomes versus others. And so I think this is where like we're just really trying to get this broader pattern. We're trying to look at that specifically. And I think getting into some of these more finer details later on is something that's very important. And especially like Lepidopterans are just very unique. You just see some very unique patterns in that clade that you really don't see in some of these other groups of insects. And so beginning to tease apart some of what's happening in that clade, I think would be really important given the results we found here and other papers where there's like just lots of reorganization in some Lepidopterans as well.
Yeah, that's great.
Very, very cool. Yeah, I was also wondering, like, what if those fusions or fissions are sex chromosomes? You know, like, can you put that in the model and somehow, I don't know, broadly see patterns? I'm not sure.
Yeah, and I also study sex chromosome evolution, so that is something that's very interesting to me. But at this moment, that's not something that's incorporated into our model as well.
Very good. All right. Well, speakers, if you'll all leave your camera on, or Kirsten, I invite you to turn yours on. We can all give a virtual round of applause. Thank you very much to all the speakers. We did our timing perfect. It's 1 minute till noon. So you guys did great sticking to time and answering questions. And I've really enjoyed all the talks today. Brad, do you have closing remarks?
Oh, outside of just thanking everybody, and obviously thank you, Lindsay. You did not get appropriate props yet for all the hard work you've done in the background, and all of our speakers, obviously. So the unsung heroes and people up front, obviously appreciate everybody coming out and taking time out of their schedule to listen to these important and very intriguing talks.
I also want to remind people that if they popped on late and missed something, or they saw a talk, they know their colleague will have, all these are recorded and they'll be up fairly soon. So people can view these later as well. With
Well,
that,
thank
I
you,
guess
because
thank you and goodbye. And speakers, if you want to stay on after, we can chat for just a second. All right.
Thanks,
you, everyone.
everybody. It's been awesome. And yeah,
Bye,
let me
thank
chat for a
you.
second.
Thank you.